Welcome to Biotanemy. Hello everyone. Today we will see the technique called PCR or polymerase chain reaction. It is a common laboratory technique invented by an American biochemist, Kary Mullis, that is used to make millions and billions of copies of a particular region of DNA. The main goal of PCR is to have enough of the target DNA region that it can be analyzed for some other purpose. For example, the purpose of sequencing, cloning, etc. The important requirements of PCR are: 1. Taq polymerase. Taq polymerase is a polymerase enzyme isolated from the heat-tolerant bacterium named Thermus aquaticus. But the Taq polymerase lacks proofreading. Instead, PFU polymerase is used nowadays. 2. Primers. A primer is a short nucleotide sequence, about 18 to 24 base pair length, which will provide a starting point for DNA synthesis. 2 primers are required for PCR reaction, forward and backward. Both of them bind on the 3' end of the 2 DNA templates through complementary base pairing. And longer the primers are, less efficient they will be during the annealing step. 3. Magnesium chloride. Magnesium chloride is added for the availability of magnesium ions, which are essential cofactors for the polymerase enzyme. The low concentration of magnesium chloride will result in weak amplification or complete failure of PCR. Too much magnesium chloride will lead to nonspecific amplification. 4. SYBR green dye. The addition of dye will make it able to track the running of nucleic acid in the gel, and also SYBR dye will function as an intercalating agent and emits detectable fluorescence when it bound to double-stranded DNA. 5. dNTPs. This consists of 4 basic nucleotides: dATP, dCTP, dGTP, and dTTP. They are the building blocks of new DNA strands. The process of PCR will be done in 3 repeated cycles. They are denaturation, annealing, and extension. Step 1. Denaturation. Denaturation is the step where DNA templates melt and separate from each other. During this step, the temperature will be brought up to 94 degrees Celsius. This temperature will vary with the GC content of the template. Step 2. Annealing. In the step of annealing, the temperature will be lowered to 56 degrees Celsius for the primers to bind with the DNA template. Step 3. Extension. The temperature here will be again raised up to 72 degrees Celsius, the temperature at which the Taq polymerase is usually most active. During this step, the Taq polymerase will add nucleotides to the primer and amplify the DNA template. These cycles will be repeated for 25 or more until you get enough amount of target DNA sequence. The results are then viewed using an agarose gel electrophoresis. Now, the PCR mix will be prepared by: 1. The DNAs are isolated from the cells using the previously given methods. 2. The PCR mix is prepared in such a way that it should contain 2/5 DNA template, 1/5 forward primer and 1/5 reverse primer, 1/5 distilled water, and the rest contain the master mix with polymerase, SYBR green, magnesium chloride, and dNTPs. Don't forget to mix the solution using a PCR mixture after the addition of each chemical. 3. Keep the PCR mix in the PCR machine and run the program. That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.